So regenerative medicine goal is try to replace those tissues or those cells that get damaged or are lost with time. And this can be done in principle by different methodologies. I have depicted here an old designed by one of the most known embryologists done 50 years ago, Waddington, where you can see a hill, and at the top of the hill is this cell that start to go down and differentiate. Now, this process can go back, and it can go all the way back in a process called reprogramming, and this has been demonstrated nicely about 5 years ago by the group of Shinya Yamanaka. It can also, this cell can, instead of going all the way back, it can go just a couple of steps backwards, not to an embryonic-like cell, but to, say, a progenitor cell, and this is called dedifferentiation, or could directly go from one valley to the other and from one cell to another cell type in a process called transdifferentiation. So I'm going to go through with you and show you a few of these processes and examples. And in particular, I will concentrate in cardiomyocytes and vascular cells. This is a scheme of the 3-4 processes, and let's go to the first one, the idea of reprogramming. So the idea is that, as you very well know, is to obtain this differentiation, this reprogramming back to a pluripotent-like state, and then from there start to differentiate your cells to the particular cell you are interested. This is a process that has been mastered in the last few years. We have developed this in a very simple assay, which is just taking one hair, obtain the pluripotent cells, and it's not an invasive procedure. It can be done very quickly. It's perhaps the methodology that works more efficiently, the obtention of keratinocytes, and from there the IPS. Now, once you have the IPS, you then, and specifically with the help of all the knowledge that has been generated in the last 2 decades in developmental biology, try to play around with the factors that appear to be critical in development for obtaining that specific cell type. In the case of vascular progenitors, after we obtain the IPS, we start to play with different combination of these factors and with different media. And here I'm showing you the results of a protocol, quite efficient protocol, because up to 70% of the cells that are obtained are vascular progenitors that give rise to both endothelial and smooth muscle cells. To the left, you see some antibody staining. To the right, you see some mRNA expression indicating the nature of these cells. Regarding functionality of these cells, we have started to put them through different assays, like the LDL uptake assay, the capillary formation assay, and injecting also then in animals, and you can see that both from IPS or ES, that these differentiated cells can after injection in mice, can connect to the previously existing structure and seem to be forming vessels. Fine. Now, the idea after the IPS was, with the knowledge, as I mentioned, of developmental biology, was to go through these mesodermal progenitors. It was okay, as I mentioned, to go through and obtain these endothelial and these vascular cells in general, but as you can see here, if you take either from CD34 or CD31 cells, and you then try to look for markers, putative markers of cardiac progenitor cells, you don't obtain that much. So obviously there is a problem with that particular lineage. The protocols we have been trying are rather, the reproducibility and efficiency are rather low. And this is not only a problem in the quantity, but also in the safety of these cells. Because if the protocol's efficiency is low, you will have to have these cells in the Petri dish for a long time, and this could be a concern for tumorigenesis. Therefore, we do need other approaches to try to get cardiomyocytes and enough cardiomyocytes and high quality cardiomyocytes. That's what I'm going to do a detour for the next 10 minutes and try to guide you through one of the approaches we are doing towards this goal. And this is by looking at nature, how nature normally does this, how cardiomyocytes are generated. What you see here is the champion of all regenerators, is this Mexican axolotl, which is an incredible animal. Whatever you cut, it grows back, provided you don't kill the animal in the operation table. You cut an eye, you cut a limb, you cut a piece of a heart, and it grows back. It is amazing. I thought it was interesting to show you a video, for instance, of a limb, of how this happened. You cut the limb, and the stump starts to grow there, and in approximately 5 to 6 weeks, this animal puts back the limb, and it puts back the limb the same size with the same number of digits, the same length, and it knows how to stop. That perhaps is even more important than the how to start. So we entertain the possibility of working with this animal for some years, but I have to confess that we haven't moved forward too much with it. Not much is known about its genetic. It's quite difficult to handle it in the lab, and we are not that many people working on it, and that's a critical mass, as you know, always needed to make some advances. We know a little bit, but not that much. But in the meantime, we thought, well, why don't we move to another model? And this model is the zebrafish that you know much better. About 7, 8 years ago, we showed that these animals also are able to regenerate their hearts. Now we have focused in other structures like the pancreas, the eye, the fins, and they also regenerate all these structures. So posteriori, I would say that this is a good animal to study regeneration. In the case of the heart, the first question was, how do these regenerate here take place? Does it come from a progenitor cell in the remaining heart, or is it a cardiomyocyte derived regenerate? For that, we had to develop the tamoxifen system that you are all familiar with, so that after adding tamoxifen, we have our cells green. And the experiment was very simple. So all what you do, you add tamoxifen, cut the heart, and then you have a couple of possibilities, that the regenerate is green, which it will indicate that the regenerate is coming from existing cardiomyocyte, that the regenerate is not green, which will indicate the progenitor existent, or you could have a mix of both. I'm not going to go through the details, just to let you know with this image that all the cells are green. So you can see here in a zoom of the heart, basically this is the fibrin clot that appears after the cut, 7 days, 14 days, and in 1 month is totally gone. And you can see that all the cells are green, that indicating that all cardiomyocytes derive from existing green cardiomyocytes. Now, this certainly give us a tool to start to understand the cellular and molecular process of how these cardiomyocytes are formed in the zebrafish. The model we have now currently working is this one here. After the cut, you have a slight dedifferentiation. This you can see in these micrographs. You see still this characteristic sarcomeric structure in some parts of the heart. And after this slight dedifferentiation, cells proliferate and start to differentiate again. Fine, so using this as a model, we have started to look at the genes involved in these early phases of regeneration. We have uncovered many genes, and we are focusing right now on them. For instance, here you have one of them, polo-like kinase. You can see that they start to accumulate here after 3 and 7 days of the cut. It is expressed near the border where the cut takes place. And if you downregulate this kinase, as you can see here, regeneration then is impaired. So like this one, we have different genes that appear to be involved in the early phase. Now, we heard yesterday by Mauro of the importance of microRNAs. We then decided to see if they were involved in this process, and obviously they are. So we have a look at these microRNAs, and we have focused on the criteria to select the one we are going to work with. Is basically that they are conserved in humans and that they have, in principle, a biological function that is of interest for the process, like they are involved in proliferation, transcription, etc. Now I'm going to show you just a couple of examples. We have many of them, and I think, and I was just briefly commenting this with Mauro, it will be of interest to share this knowledge between zebrafish and human and see which one of them are really conserved and try to, from there, try to understand better the human process. So for instance, this one, MIR99-100. The target proteins for this MIR, they seem to be involved in different processes, but mainly in organogenesis. They have some role in chromatin remodeling, which we have an interesting observation I will not go through today, but the importance of chromatin remodeling in regeneration cannot be understated. I wish I could have some time. We'll see. I have a few slides at the end, and maybe I could give you some examples of this. In any case, we have taken some of these microRNAs, and we have looked how their expression while regeneration takes place. So here to the left, it will be the control heart, and you can see that some of them are upregulated after 3 and 7 days when the dedifferentiation process takes place. We have started to play around with them to administer to the zebrafish, and we can see that high levels of all these ones that we have taken produce scarring after amputation, and regeneration doesn't proceed normally. I don't want to go through the details. I just want you to leave with a message that in functional assays, these microRNAs seems to be active and abolish regeneration. And also their targets. So if you take some of the targets of these microRNAs, the same process. So they are able to inhibit this process of zebrafish regeneration. The general idea is that in the zebrafish you have two phases. The first phase is that phase of dedifferentiation. It's a proliferation permissive phase. And then after 7 days, more or less, is when you start to have the proliferation. And if you look at just the graphic of how these microRNAs and their targets are expressed, you can see that the microRNAs are high in the initial phase. They then go down. And this is the opposite for their targets, that they are down in the initial phase and then they go up. This is a little scheme of some of the pathways we are now working on, and it's not important. What I wanted to say here is that the zebrafish model is giving us some ideas of how the process of regeneration takes place. It seems to go by this process of dedifferentiation, and it is allowing us to obtain factors that we may translate then to the human in order to try to obtain, as I said, cardiomyocytes. So let's go back now to the human and to the cells. So I mentioned that with this process we failed to obtain a reproducible and efficient protocol for cardiomyocytes. So what about dedifferentiating the cells a little bit the way that the zebrafish does and see if we can be more efficient? And this is what I show here at the bottom of the slide. So what we will do is try to introduce factors, for instance, the reprogramming factors, that make the cells a little bit plastic, but you don't obtain really iPS cells. It's at that moment when you start to introduce the key factors for differentiation. So I think this slide shows better what I want to say. So we have now in the case, again, of vascular cells. So we have your initial cell type, and then you add these reprogramming factors so that you destabilize the structure of this initial cell, but you don't leave them there for a long time. Instead of obtaining iPS, you just have the factors for a few days, and it's at that moment when you start to add the key factors and the media that will give you the specific cell type. Here is an example for vascular cells, both for endothelial and smooth muscle cell. The protocol now becomes much faster. You don't have to produce iPS cells, even more efficient. In this stage, you don't see the markers that will characterize an iPS cell state. And this is important because safety at the end is a concern in the generation of iPS cells. So if you avoid the iPS cell state, that may alleviate this concern of safety. And it's in this second step when you start to add your factors. And for instance, in the case of vascular cells, we just need to add one factor, in this case, SOX2. So after we have made the cells plastic, just with one factor that can even, we know now can substitute this gene biochemical, you can obtain your endothelial or smooth muscle cells. So I think this is an alternative approach to iPS generation for the different advantages that I have mentioned, but perhaps the key one is here, is that after this procedure, and contrary to what is seen with iPS, we don't generate any teratoma. And that's obviously, from a clinical point of view, will be important. So we have now start to apply all these call this procedure towards cardiomyocytes. And so we are now starting to add all these proliferative factors that are conserved from the zebrafish into this procedure. We are also doing the same thing with these MicroRNAs. It's the initial phase of the experiment, so I don't have too much to add at this stage. But I would like to insist on something that was mentioned yesterday, and even though it is not very well connected to the topic today, is the fact that in order to be able to compare how our cell products are really functional, and that's due to what we really do in the lab, we need to go through matching the cell lines we are working with. And for this, I'm going to mention just this example. These kids that have a mutation in lamin A, and that they die prematurely around age 10, and the main cause of their death is atherosclerosis. Now, this mutation is due to altered splicing that give rise to a different form of lamin, a shorter form called progerin. And the accumulation of progerin is what caused this phenotype. Now, the phenotype is a rather complex phenotype. The accumulation of progerin gives rise to many nuclear defects. So what we have done has been to generate iPS from these kids. And as you can see, in these iPS, neither lamin, the wild type, or the mutant form, progerin, are expressed in the iPS state. And this is great because then it allows us now to start from iPS that progerin is not present to start to differentiate that to vascular cells and see how the accumulation of progerin start to alter this process of differentiation. So we have done this, and the problem we face is we start with this mutant cell, we get iPS, progerin is not there, and now progerin start to accumulate, and let's study the process in vitro. But to what do we compare this process? And I think this will be a problem for all the community. Do we compare this to another individual and do the same process? Probably we will get some insights, but the genetic background of that other individual will confound certainly the results. So we need to have a situation where we don't have this mismatched genetic background. And yesterday, and that's the reason I put these slides, there was a couple of talks in gene therapy. And I felt it was important to just say a couple of words on the importance of having a situation where the only confounding factor is your mutation. So instead of any cell here from another individual, or even the same individual, you should have a cell that the only different is the gene you are working with. So we did a correction of the gene, or we even generated ES cells where we have introduced that particular mutation. Now you know very well that in the mouse is relatively straightforward. It's not the case for humans, but recently with the advance of zinc finger nucleases and talent, this is being possible. We have developed a different approach, which is the helper-dependent adenoviral vectors. And I have to say that we are very excited with this technology. It is incredible efficient. So you can see here different loci that we have targeted. And there are cases that you get efficiencies up to 100%. If you compare this to the zinc finger or talent that is below 1%, you compare this to the fact that in the case of the zinc finger nucleases and talent, you have off-target effects. Certainly, this will be an advantage. Moreover, you can insert large pieces of DNA, and importantly, you don't need to focus just on pluripotent cells, but these virus are able to transduce all the mesenchymal cells that we have been working with. And you can, of course, introduce your knock-in, etc. Hypoxia, we have put the fish, we have developed a system to put the fish in under different oxygen concentration. We have cut their hearts, and then they look at how they regenerate. And the message is very simple. So hypoxia helps in promoting cardiomyocyte proliferation. You can see just the bars. Here it will be in blue, hypoxia, normoxia, and then hyperoxia is totally the opposite. It promotes cardiomyocyte differentiation, and when you block HIF-1 alpha, then the process is also altered. We have now obtained fish where there is not just a dominant negative, but a knockout of this factor. And I wanted to put this, we have many more data on this, but I wanted to put it there because, as I said, I have always been intrigued about the role that hypoxia has in human cardiomyocyte regeneration, and I thought I will try to get some insights from you. How could we try to interpret these results of hypoxia in the human context? This work was done mainly at the Salk Institute in La Jolla in California with the help of Chris Joplin in the Center of Regenerative Medicine in Barcelona. This is my lab. I will not specifically pinpoint any person in the lab. This is a team effort where everyone is doing their best in producing some of the data that I have introduced to you today. Thank you very much for your attention.